Okay, I'm sorry for that. First of all, I thank you for introducing me. It's really a pleasure to be here. This morning, we all have some connections with Switzerland. I'm not born in Lugano, but two of my daughters, out of my three daughters, are born in Lugano. So there is a connection with the place here. I'm really pleased to speak after the introduction in pharmacology by Professor De Sarro, because I think indeed this is a conference and we have also, let's say, a sister or a brother activity where we look across disciplines. So it's maybe strange with this title to speak about in a session on pharmacology. I will be speaking in next generation models and methods for understanding health implications of chemical and biological contaminants in food. But I have a medical background. I'm from training an engineer, biotechnology, but I've been following an education in the disciplines of medicine. So I've been working in multidisciplinary teams with many companies, many academias, many regulatory authorities, and exactly that multidisciplinarity we feel here. So I've been working also with many organizations globally, and I think this is the spirit of coordination and collaboration we have seen. We started to talk about efficiency and pharmacology and looking when you have the window of a medicine that functions. But I was always in the company where I started in Belgium, part of the bad guys, because I'm a toxicologist. Now, in the beginning, indeed, we have been seeing also in the presentation, in preclinical testing, you start often to work with animal models. I've been working and dedicating 30 years of my life to embrace more technology to fill that gap, that we don't have to wait until pharmacovigilance to see adverse effects, and look if we have now better methods and models to predict early on adverse effects on people. So we are talking in pharmacology on personalized medicine. I'm talking a bit on the gaps in personalized toxicology. So I come from the area of tissue engineering, and this is from of these tissues models. So that is the topical toxicity, but we also talk about systemic toxicity. Now, how does that link with regard to the area of food? Because we have been seeing that collaborations for pharmacology are with our European Medicine Agency, EMA, but also on these new technologies and new methods, our Food Safety Agency in Europe is working a lot. Again, this is a global market, so we collaborate because we have a big societal challenge. By 2050, the planet has to be feeding 10 billion of people. We talk a lot about green transition. We talk about alternative therapies. There is a big issue if you want to go to things that are sustainable, green chemicals or green pharmaceuticals, that there might be a trade-off in terms of safety. This is not only for the food sector, but it is also for the pharmaceutical sector. I have a collaboration agreement on green pharmaceuticals. So that's why I'm speaking a little bit about the multidisciplinarity also across the sectors, which is very good to look across the different sectors to learn from each other. What we need to do now, and these are 2030 targets, is obviously within the field of food, sustainable food system. They say you get down, for instance, the percentage of antimicrobials. We need to look at alternatives for that. We need to get rid of the pesticides. Yeah, we need better nutrient food, and we need more organic farming. But that means also that we need to look at other types of therapies. And we just saw already what type of therapies we have. And also in this field, we look at a new generation of antimicrobials. We go also lots in botanical essential oils, and we also look at biopesticides. But the same transition happens in the pharmaceutical industry. So actually, tomorrow at the European Commission, we start with a conference on One Health. That means health for everybody. Health for the people, the kids, the elderly, the different communities, health for, let's say, the animals, and health for our planet. So One Health is now the buzzword, I think, across our disciplines. We have been seeing in the previous presentation that worlds are very close to each other. So we have been talking when a drug enters the body, we want to know two things about a chemical. We want to know what the body is doing with that chemical or pharmaceutical or with that contaminant, but also what the chemical or the pharmaceutical is doing with the body. So one of the big things is that we need to know about the kinetics and dynamics of substances. Metabolism, ADME, as just was presented by Professor Di Saro, is very important. We need to know about the absorption, the distribution, the metabolism, and excretion. Now, obviously, in the field of metabolism, there is a huge species difference. So our body is dealing with chemicals different than some of the animal species. I started to work in the field of liver transplantation to get some pieces of human liver cells to start to look at the difference in metabolization between animal species and between humans. We also started to look at differences in individuals, so polymorphism. These methods, these new approach methods, need a lot of standardization because cell and tissue culture are prone to biological variation. So I've been working 30 years in standardization of new technologies, cell and tissue culture technologies, to get them now embraced also in discovery, in preclinical research, and also then in the overall branch of many disciplines. So 20 years of work to look at the kinetics and the dynamics and to look at tools, how we can go better, not only into personalized medicine, but also in personalized toxicology. A little bit the gap that Professor Di Saro spoke about. Many work, many collaborations. So today, with all that pre-work in new technologies, both in vitro, cell and tissue culture models or subcellular fraction, or in silico mathematical models to, in a way, puzzle back the information into the different subpopulation. Where are we? I liked a lot the introduction, and I give you an example of something that went wrong in the past. We have many of our European cohorts that are European cohorts, but many of the people that might suffer are other subpopulations. So can we do something to have also medicines for different subpopulation? I'm biased because I'm Belgian. This is an example that shows in, I think in Italian, it is lepra. Leper, you know about the disease in Hawaii. It has been executing quite a whole population of Hawaiians. Father Damien, who was Belgian, who went there to treat the people. Basically, this was a population which had genetic susceptibility to lepra. So with the Human Genome Project, we have much more information, but we have to be careful that we understand what we can do with Human Genome Project, and that we also take care to see that our technologies are developed for the susceptible population. We talked about children and we talked about the elderly, but we have many diversities in population and many genetic diversity. So also in drug development, but also in toxicology, these are the areas we have to focus on. I can very well talk about this issue because in 2015, I almost died. Next to me, some of you might know what happened to my friend Sylvia. Sylvia took thalidomide. Thalidomide was the drug that was given to pregnant mothers, and most of the kids, they were born without the limbs. Sylvia's mother took it later in the developmental stage, so she got still the limb formation. This is many years ago. Thalidomide was a drug that was tested on animal species, and when it went into the market, it gave these problems. I myself, the little photo there is me in 2015. I almost died taking a drug, a good drug, montelukast. It's an anti-leukotriene. Obviously, is the drug bad? No. But I'm a susceptible subpopulation. This drug has been given on the market later. So pharmacovigilance is a good way to to pick up adverse effects, but we need to do better. We don't need to go in the market without knowing the background of some drugs. We talk in the other aula about quantum computing. In life science, with quantum computing, we can do a lot. In the previous lecture, we got some marvelous examples how we try to work in the field of cancer and other disciplines to get better personalized medicine and also other personalized toxicology. I will not talk about cancer. I will talk about another effect of compounds on the human body, which are targeting all our glands, our hormones. As endocrine disruption has been known in wildlife many years ago with the crocodiles that were, let's say, becoming feminized. I've been working the last part of my career on endocrine disruptors. Because for endocrine disruptor testing, we don't have good animal models, and these compounds need to be tested. So over the last years, I've been working together with OECD to check if we have better predictive methods to deal with estrogen-mediated effects, androgen-mediated effects, thyroid-mediated effects, and effects on steroid production. We don't have good methods. So when you look at let's say the signaling pathway of endocrine disrupting chemicals, and for instance, that target action in the hypothalamus pituitary thyroid axis, you see that many of the compounds on the left panel, I'm sorry, I use this, these are all list of some kinds of endocrine disruptors. They interfere with signaling pathways in our body at different stages. So they either interfere with thyroid hormone synthesis, when I take the example of thyroid disruption, or with thyroid hormone action. So we are developing with new technologies, many new methods. We cannot do that in isolation. And Professor De Saro said, you need a collaboration between developers of methods, between industry, between regulators, between companies, small and big companies. And you also need to embrace new technology, like we're discussing in the other room, artificial intelligence and quantum computing. So one of the last projects that I've been leading was a project where we put all heads together, the developers of methods, but also our companies, big companies and small, to come up with a battery of methods to better predict adverse effects on the thyroid system. Last but not least, why I'm here today, you would wonder, because what I've been learning for the last 3 decades of my life, when COVID-19 started, the kinetics and the dynamics you can do with chemicals, you also can do them with pathogen. So together with some people here in the room, we have been working on a book chapter of this book, and for those, I brought it with me, which is entitled The Coronavirus Pandemic and the Future. It's edited by the Royal Society of Chemistry, on a chapter that shows that multidisciplinary teams are the magic of today. So we put together people from different disciplines to write a book chapter on knowledge from human relevant cell, tissue, and mathematics-based methods as key tools for COVID-19, just embracing the science we have been learning over the last years. So these are some of the work we have in the book. So we use the new technologies there. I don't want to go in detail for the sake of time. So this is why I'm here today, because in our book chapter, we start, besides embracing alternative methods and models, to also say that alternative therapies like diamagnetism as an alternative or integrating cellular therapy for COVID-19 has potential, and we have to look together in that. As a bit this morning said, we are all on our own, but together we are strong. So I think we are not a drop in the ocean. We are all together, the ocean in a drop. And I think these are some things. Science impacts us all, and it's complex and dynamic. This type of conferences, and I'm very grateful for inviting me here, these type of conferences make a difference. I'm very happy to speak between 2 people from the University Magna Grecia, because last week we just signed a collaboration agreement between the University of Catanzaro. So I'm looking forward for the rest of the conference, and I'm really happy to discuss any further things with you all. Thank you so much for inviting me to the organizer. It's really a pleasure. Thank you.